Well, good morning, everyone. I'd like to welcome you to Proteoform Thursday, our monthly seminar series, trying to advance the understanding, the enjoyment of top-down proteoform biology and all associated things. So thanks for coming today. We're fortunate to have Aaron Bailey with us to talk about some of his work on intact proteins. But let me first give a little quick introduction, a little news on what's going on, and then we'll jump into Aaron's seminar. So the consortium, as you know, there's two main efforts we do. One is to try to build the community, build the knowledge, education and training, workshops, collaborations, and really knit the community together because we know there's some challenges out there and only together we're going to get these figured out. The other is to try to guide the technology and help develop protocols, methodologies, and develop the proteoform atlas as we can all take advantage of that. So if you haven't seen it yet, the proteoform project paper has been out there for a couple months, and it's two main thrusts around this. One is to build a set of human proteoforms in the atlas that we all can take advantage of. And the other is to get to that point, we're going to have to develop new technologies, new approaches, things we haven't even attempted to do yet. So there's two main thrusts to that, and so far the interest has been quite good. Paper's been downloaded over 15,000 times. So if you're one of those 15,000, I hope you enjoyed it. If not, then you can go ahead and grab it. It's an open access paper. The main thing coming up is ASMS. Many of you will be going or participating in this. There's a couple of things I just want to mention. One is there's two new courses available for top-down proteomics. And the key thing here is the registration deadline is next week. So if you're interested, you need to kind of get on that. There's a top-down proteomics course being given on Saturday from this esteemed panel here. It's going to cover all the aspects of top-down. So if you or a colleague are interested, go ahead and jump on that. And the other is to address the data analysis side of it. And these fellows put together this course, which will be on Sunday. And that's one of the biggest challenges. So that's also something probably of interest to a lot of you. And as far as activities at the ASMS, there's an oral session on Thursday. There's two poster sessions. Then there's an evening workshop on Tuesday on top-down mass spec, which would be kind of interesting, more of a community discussion, wrestling with some of the issues thing that's open to everyone. And on Wednesday, there's a workshop on Fred McClafferty's career. He's done a lot more than top-down, of course, but top-down will be a piece of that as well, I'm sure. So those are some things to look at for ASMS. So next month, I'll let you know that Moe Zhao is going to be speaking to us from PNNL. He's done a lot of work in this area. Be interesting to see what he has to tell us about that. So that'll be on the latter part of June. There'll be more information coming out on that shortly. So if you're not a member yet, please go ahead and join the community. There's no charge to join and be connected with us all. We'd love to hear your input and have you connected to what we're doing. So this wouldn't happen without the support from our major sponsors, Thermo Fisher, Brooker, and SciEx, who are very appreciative, as well as our consortium supporters, Agilent and Pfizer, that allow us to do all these kinds of things and run these initiatives. So we're very grateful to their support. And if there's anyone out there who would also like to get engaged and support the consortium, you can please contact me. Be happy to speak to you about some of the options we have for that. So without further ado, let me turn it over to Dr. Aaron Bailey. He's the Associate Director at the Mass Spectrometry Core Facility at the University of Texas Medical Branch. And he'll be speaking to us today about the development of a versatile denaturing intact mass toolkit for integrative protein structure characterization. So go ahead, Aaron, it's all yours.
Okay, thank you. All right, can you see my slides?
Not yet.
Okay, let's try this again then. All right, we're up on the wrong foot. There we go. Okay.
Looks good.
All right, thank you. And thank you all for joining today. So we're going to talk about integrative protein structure characterization and the role that we see denaturing intact mass that it plays in this structural characterization. I'm speaking today from the University of Texas Medical Branch, and we're in the mass spec facility. So here in the mass spec facility, we look at both large molecules and small molecules. We do a variety of custom work. On the large molecule side, we look at both proteomic type analyses. We also perform structural characterization for proteins, both at primary structure level as well as higher order structure. And we do a little bit of oligonucleotide work as well. Focusing on the protein portion, we study everything from networks to isoforms. And so what does that mean? We often look at whole proteomes. These are, differential quantitative proteomic type experiments. You can start to sort out cell compartments and see where differences lie. Looking at protein complexes, and we also look at ultra pure single, there's quotes around that, protein samples, where really at that point, the objective is to understand the different forms of proteins. Of course, we use high resolution separations and detection. This is mostly LCMS or LCMSMS. And the way that we do that is, depending on the sample, it's either proteomic style methods or these structural characterization methods. And you can see here in the middle in my cartoon that there's a lot of gray area, or sort of meat in the middle, where these applications, I think there's a lot of opportunity here. So this is where we play a lot in our lab. Okay, so we have this integrative protein structure spiral that I like to show a lot. And basically this is our logic for assembling all these different assays that we do. So we start simple, right? We look at, for a new protein where we need to know a lot about it and we know sort of very little, we might look at sequence confirmation initially. This is a peptide mapping style approach. Of course, this gives us primary structural information. Beyond that, we continue our primary structure analysis really with intact mass and denaturing intact mass in particular. This has a very specific role. This is, we call this and consider this the foundation of intact mass, the denaturing intact mass. Of course, it also builds on our primary structure knowledge. And then as we keep going, we continue our knowledge building in primary structure land. Again with peptide mapping, but in this case, we revisit with PTM site localization. And we can keep going looking at tertiary structure, disulfide mapping, cross-linking, and of course, native intact mass, which will give us quaternary structure information for protein-protein or protein, say, ligand complexes. I'm going to speak a little bit more here about the intact mass side of things. We have an assay or several assays which could be run in either denaturing or native conditions. So depending on the sample type or the measurement goal, ranging from sort of simple to complex with multiple polypeptides or ligands, the way that we go about this is we use a variety of liquid chromatography formats, size exclusion, reverse phase. We can also look at these samples with native means, native LCMS, again, size exclusion, ion exchange. We can do nano infusion. There's a few that I didn't list here, that we dabble in. These are kind of the main fare. And of course, your mass spectrometer needs to be tuned to the appropriate settings in order to say catch your native proteins or your denatured proteins. So size exclusion is really a handy tool in this realm, which allows us to operate both in the denatured and native realms. And in this case, we can actually use the same exact column, but just change the mobile phase. I'll get into that in a second, but it's quite handy. So the gist of our workflow is that we use size exclusion at analytical flow rates coupled to UV detection. And of course, those proteins are eluted directly into the mass spec. We're using Orbitrap Eclipse MS. And the raw data that we produce is analyzed by deconvolution. So we'll take a deeper look. For our size exclusion, I can't say enough good stuff about this Waters BEH Sec 200 column. It's a 30 millimeter column. It's a guard column is how they sell it, but it's, we call it a desalting column. That's what it does primarily. And for the denatured work, we use 30% acetonitrile, a small amount of acid, formic acid, a tiny bit of TFA. And for the native stuff, we typically use 50 millimolar ammonium acetate. In this case, I'm showing you an example of 8 traces overlaid with the denaturing sec desalting column. And, it's identical. So this tells you that it's a robust, very reproducible method. Getting into the mass spec portion, of course, we use LCMS, and this is all glued together by electrospray. where we produce a series of cations for every protein isoform. And the deconvolution analysis is what allows us to take that redundant type of information and then convert this to a neutral mass or zero mass and zero charge mass, where in this case, we're looking at a small protein, concanavalin A, and we see some trace amounts of 2 glycosylation or glycation, excuse me, and also 1 glycation. So this is roughly how we do all of these experiments. Maybe I'll just add the deconvolution that we prefer is this biopharma finder, where we can use the respect algorithm for average mass deconvolution. And this is kind of accompanied by the sliding window algorithm, which allows the deconvolution to be done in a time resolved manner, which is really well suited for LC or other separations for intact mass. So kind of getting into the heart of the talk, there's something that we've been working on that we call full scan PTCR. And the message is that there's really 3 different aspects where this approach can help you for denaturing intact mass. So it's a similar approach as we just saw. We're using denaturing mode for our SEC-UV. And then those proteins are eluted directly in the mass spec, or we trap eclipse again, at which point we perform a routine involving this protein. This is an intact protein here, kind of beads on a string with these cation amino acids here. And then this is allowed to react inside of the Orbitrap eclipse, not in the Orbitrap per se, but in the instrument with these reagent anions. And the 2 react and produce a charge-reduced version of the proteins. Of course, those data are still deconvolved using sliding window. This work was just published recently in Analytical Chemistry in February. So we're still pretty pumped about that. And so the outcome of this is where you have a intact protein, particularly in denaturing conditions where you have some spectral crowding. Pretty often we can reduce the charge state with more reaction time, and it produces higher M/Z species. Again, there's 3 objectives or challenges I think that are overcome or mitigated using this technology. And while these are interrelated, I think it's helpful to break them out into individual categories as we're doing here. So overcharging, micro mixtures and macro mixtures, otherwise micro heterogeneity and macro heterogeneity. And in my cartoon, I'm trying to show that there's actually different utilities for each of these different situations. And sometimes you get effects from both of these. Before we get into the applications, I guess it's important to talk about how this is implemented in the Orbitrap eclipse. In our report recently published, as you can see, we kind of list all the various steps, and that's worked out here in the cartoon, but I'll give you the quick version. So here we have a protein, intact protein cation, polycation, and those proteins are eluted from the column, they're electrosprayed, and then they're isolated in this ion trap. Next, we have our PTCR reagent. I guess whether I mentioned it or not, this stands for protein transfer charge reduction. So this charge reduction reagent here, C14F24, is produced in the source itself, in a separate part of the source. And then those anions are basically sent to the ion trap where the anions and the protein cations react. And then those proteins are analyzed in the Orbitrap where we can produce these spectra. One more thing to talk about in terms of the technology. So I mentioned in the title of the talk and a couple times already that our platform we consider to be versatile. So one important aspect of that is in the size exclusion chromatography itself. We really operate this in kind of 2 discrete modes. The first is this what we call screening mode, where we're operating the flow rate at 200 microliters a minute. This is a constant flow rate for a 4-minute runtime. This allows us to get about 20 scans at a resolving power of 15K. And so about 12 seconds of analysis time where the protein is actually eluting. You can see with size exclusion, of course, the small stuff comes out later. So we get very, very nice separation between the intact protein and the buffer salts. There's a lot of literature on the fact that this is either the most or one of the best efficient desalting techniques that there are. It's also very low absorption. We can't say enough good things about size exclusion. The other cool thing is that it's very simple to slow down this flow rate and do this detail mode, which we call it, where we're optimizing now for sensitivity and resolution. In this case, we slow the flow rate for a portion of the run to 20 microliters a minute, and the total runtime is 20 minutes. So of course, during the 20 microliters a minute time frame, everything comes out slower. We're getting now 250 scans at a resolving power of 50K. And this is about 3 minutes of protein analysis time. So it allows us to do some different tricks. So understanding initially how PTCR should be implemented and optimized, we set about simply using the ion trap because it gave us more range at which we could isolate. So using the ion trap to isolate different widths of M over Z space, 3,000, 500, 100, you can see relative to the MS1 spectrum of 20 nanograms of BSA intact protein, what we found is very clearly that using these large isolation widths in the ion trap, we're able to get the most signal or bang for our buck, the most signal both in raw spectra, signal to noise, as well as the deconvolution signal measured in peak area here. So this 3000 M over Z isolation or broadband isolation, we're calling full scan or fsPTCR, where you isolate the entire charge state distribution and then perform PTCR. Now, understanding how much we should react is kind of the next order of business. And we did these experiments again in our screening mode. We can do this in triplicate. These are 4-minute runs. So in a couple of hours, we've got a statistically significant data set at a variety of different reaction times. And in doing so, you can see that 0.1 milliseconds, essentially 0 charge reduction, moving all the way out to 10 milliseconds, where you've now started to push ions off the page. And in this case, we find very clearly, again, plotting our signal to noise in the raw domain, as well as deconvolution peak area, we find very clearly that at the point at which you begin to overreact, that is, send ions out of the scanning range, you really begin to lose all your sensitivity. So that's happening somewhere between, it looks like, this 4 and 6 milliseconds. And so, the objective, the idea is to not overreact, given a pretty hard and fixed 8,000 M over Z mass range. Okay, so looking now and comparing the MS1 to an optimized PTCR reaction, we're still looking at BSA. This time it's 200 nanograms. And we're looking at the raw data here, where you can see at high pressure, like we typically operate in these experiments, we're generating what appears to be resolved ions out past, a little bit past 4,000. If we look at the apex charge state at 31 plus, we're getting a signal to noise of about 60. And we're able to deconvolve several species. So, we can tell that the protein is there. There's a number of species that we can't identify, but there are some that are clearly identified and substantiated by the peptide mapping results. Now, doing this charge reduction experiment, we've reacted to the point, I think this was 6 milliseconds. And so, we're right at the edge of the spectrum. And when we look at that apex charge data plus 12, we see we get about a 50% increase in signal to noise. So, that's good. What's interesting is actually the deconvolution results suggest that the data are maybe even better than that, where we have what looks to be a greater usable dynamic range. And that's really important, right? It's really about how much of this you can use. So in a protein that doesn't really exhibit any heterogeneity or structural weirdness, BSA with charge reduction is already giving us better data. So that's a good place to start. The next piece of this is that we have, we then reduced fully BSA, and BSA has 35 cysteines. It's actually 17 disulfide bonds and a free cysteine. So all 35, and this is not trivial, you kind of have to force this biochemically. And in the MS1 data, we find that this fully reduced BSA produces very highly charged species in excess of 100 plus, as well as a higher mass portion of the spectrum, which does not deconvolve. It's considered noise. We thought that this was fragmentation initially, and it turns out we can't deconvolve any of it. So it's some kind of noise artifact in the signal processing is what we think. All right, so the raw data, if we look at this, we do get a clean signal, or a clear signal, I should say, at the 53 plus, at about 61, 62 signal to noise. And we can deconvolve this spectrum, and we get 2 species. All right, so when we charge reduce this, the improvement is definitely more dramatic than the MS1 comparison at the denatured BSA, like I showed in the last example. So in this case, we have this reduced BSA, which is roughly a third of the overall charge state as the initial MS1. Our signal to noise is improved by about 3-fold. And now we're able to produce a deconvolved result, which shows very clearly some additional adducts. We don't think that these are ESI non-covalent adducts, but rather nitrosylation is the best candidate for this. It's an exact fit, though the peptide mapping is tricky to find nitrosylation. So what this got us thinking about was, okay, what's the difference between non and reduced BSA, right? We have these 35 cysteines involved in disulfide bonds. And when you remove those, we have this structure now, which is presumably more denatured than the quote denatured intact BSA. So, we see this greater increase of signal to noise when we use this PTCR technique. So this gets us into the first real finding, I guess, of our study, looking at using full scan PTCR to address overcharging, which occurs in unstructured proteins put in denaturing conditions. Now, these are not synonyms, right? Unstructured proteins then placed in denaturing conditions. So why is overcharging such a big deal? Well, some folks, the Matthew Bush lab at University of Washington showed a few years ago the charge state, and other folks have shown this as well, but I like this example because basically they used BSA, which we also used, and they showed that charge state is directly correlated with collision cross-section size or area. In this case, we see the collision cross-section area plotted on the y-axis and then charge state on the x-axis. And you see that they measured various charge states available and produced by these native intact BSA experiments, denatured intact and supercharged reduced BSA. So it's actually several fold larger comparing to the native and denatured when you've unraveled this protein. And the Broadbelt labs showed something similar happens inside of the Orbitrap, actually the same year, 2018, and where they showed that pressure plays a large role in this. But basically, the larger the higher the charge state, of course, the larger the collision cross section, the greater the decay constant. So the steeper the transient decay from the Orbitrap signal. And what this means then is that overcharging comes from these extra large collision cross sections, and those basically generate steep Orbitrap transient decays, which then, of course, leads to poor OT sensitivity. So we think that's what was happening in the reduced BSA. And then remembering this and kind of understanding this further, we applied this to a protein which came through our lab in collaboration with the Gagnon lab here at UTMB. This is initiation factor 2. This is a 91 kilodalton protein, which is partially unstructured. There's a flexible region here that sort of sticks out in this protein. It's not even fully unstructured. However, when we start to look at the MS1 data, it's not usable, really. We get a similar thing, but maybe more exaggerated than the reduced BSA, where we have an appreciation of very high charge states here, well over 100. And then we see in the high mass region, the noise and the non-deconvolvable signal, right? So some kind of artifact there. And then if we take a zoom in of the apex charge state there, we get a signal to noise of 34 that was measured. And the deconvolved data are not usable. Basically, this protein was shown by gel to be essentially mono disperse, highly pure. And even so, we got a mass accuracy of 712 ppm less than the theoretical mass. When we do the charge reduce analysis, we get this beautiful, it looks like a totally different protein. I assure you it's the same one. Beautiful charge reduced intact mass spectrum. The 23 plus here shows an 11-fold improvement in signal to noise versus the MS1 result. And now we're only 37 ppm off from the theoretical mass. So this is a massive and very dramatic improvement. And I hope that this goes to show you guys that this overcharging is something that could be overcome. And these difficult proteins actually can be analyzed in denaturing conditions. So moving on, we're going to look at the second example or really application for full scan PTCR. And that's improving the quantitation of intact proteins, or just generally the sensitivity of intact proteins in macro heterogeneous mixtures. Okay, so we start this off by looking at an artificial 7-protein mixture that we have. And it's basically an equal molar mixture of BSA and 6 other proteins here from 14 to 140 kilodaltons. And then in the second mixture, we've diluted BSA 10-fold. We basically find that PTR improves the sensitivity of all the proteins up to about 130K, but it does not help trastuzumab. So let's look at the data. All right, in this case, this is the raw data, and we have up top the MS1 and below the charge-reduced PTCR. BSA in this case is indicated here. It's at the low M/Z kind of high charge region. Trastuzumab is visible, but once we charge reduce, trastuzumab is sort of again overreacted off the page, and we can see BSA pretty nicely here. In the 10-fold dilution, we still see BSA, but it's much lower, and it's also lost in the mix here in the MS1 result. But so it's here, and I'll show that to you in a second. The deconvolution results, basically we see BSA in both cases. Again, we're not seeing trastuzumab in the charge reduced species because it's become overreacted. We actually also see slightly better sensitivity for some of these highly modified proteins, ovalbumin and fedouin. These are both glycoproteins. And we see a better representation of the BSA dimer as well, which is neat. Okay, and then we look at the 10-fold diluted species, and this is actually where it gets very interesting. So in both of these arrows here, we still see some BSA in the MS1 result, as I've indicated above in the raw data. But the problem there is that we actually are measuring almost 40-fold less, right? So a factor of 4 off from what we know to be is in the mixture. And when we charge reduce that, that corrects pretty closely to 10. So exactly where we would expect it to be. So that's pretty exciting. Of course, the other thing to consider about this, and as we study these proteins individually, we find that there is a benefit for all of these proteins, alpha-lac, beta-casein, concanavalin A, ovalbumin. I'll show you fedouin in a moment. Trastuzumab, however, is just too big. And what we see is that even at the optimum reaction time of 2 milliseconds, basically we're not gaining any sensitivity in the deconvolution. We're also, it appears, getting about 10-fold less in signal to noise. It would be really interesting to be able to do this with a greater mass range. And we think that there's a lot of promise there for antibodies when that comes around. But at the moment, we're sort of stuck with 8,000 M/Z. And it appears that this is essentially not usable. So our usable range is ending before trastuzumab. Okay, so then the third application area is microheterogeneity, where we see that we can actually identify more proteoforms. This is probably not a radical finding, but how it works is a little bit surprising. So we looked deeply at a protein that we had in great abundance from Sigma in a bottle in the freezer. And basically fedouin is bovine fedouin is what we looked at, where this protein has been studied many times. The Heck Lab has a great paper on this, and there's a few other great papers. There are a number of PTMs on this protein, 3 N-glycosylation sites, up to 6 O-glycosylation, and up to 8 phosphosites. So with this complex protein, the way that we wanted to move forward was, of course, peptide mapping the entire protein. We were able to get 99% sequence coverage with just one protease, GluC. And with those data, we plotted the relative abundances of the various proteoforms. In this case, glycoforms in this case, these are at the peptide level. And we went down to the fourth most abundant glycoform in this case, which is important when you look at historical data and what we found. We also covered all the ogly sites and all the phosphos sites and plotted the relative abundances of each. Okay, now looking at the microheterogeneity at the intact level, this is an untreated sample, so all of those PTMs are present in their own relative abundances. At the raw data level, we see that we're getting a signal to noise of 36, which is not great. You know, there's a lot of interferences here in these high mass protein ions. And the deconvolution, while we can deconvolve quite a bit, it's curious that the most intense species is at 95 ppm. When we do the charge reduction, strangely, surprisingly, we actually got lower signal to noise at around 17, 18 signal to noise. And then the deconvolution, while the signal was lower for the deconvolution peak area, the mass accuracy was much nicer. And if you look at the deconvolution results at the baseline in particular, it does appear that we have a greater dynamic, but we were really struggling with understanding why there's lower signal for the PTCR spectra. So when we take a closer look at the deconvolved data, and in the top panel here, we have the MS1, and then below this, we have the full scan PTCR data. What we see is that we plotted the mass accuracies for all of these species that were identified. We used a window of 100 ppm as our maximum tolerance. And what we found is that if we look at this most abundant species in particular, that across the board, we began to pull out these shoulders of a lower abundance proteoform. I say lower, but it's high enough in abundance to actually skew the measurement at the MS1 level. But in charge reduced conditions, we pull these shoulders out of the main species, and we find that it improves our mass accuracy tremendously. So in the MS1, we get a 95 ppm mass accuracy, but these two individual species, which are confirmed by the peptide mapping results, show that we have a -12 and a +23, so much nicer mass accuracy. And we see that across the board, that's also the case. So if we calculate a weighted mass error for all of these species, we find that the PTCR radically improves the mass accuracy, and in this case has given us double the number of proteoforms from 56 to 112. Okay, so in the last part of the talk, I just want to go over the application of charge reduction within a larger project where we're looking at the integrative characterization of a 25 kilodalton engineered glycoprotein. So I'm using somewhat generic language here. We don't have permission to give all of the details. This is a project in collaboration with somebody who I'm also not allowed to mention. But this is an exciting project, and I'm very thankful that they're allowing us to share the data as we're able to. Okay. So in this case, we're looking at an engineered recombinant protein, which is, again, 25 kilodaltons. This is expressed in Pichia pastoris, which is a yeast. Pichia is a great high yield system, making a lot of protein that's become increasingly popular for making biotherapeutics lately. The structural characterization is really challenged in this case by two main features of this protein, where there's proteolysis or truncation at both termini at once, or potentially at once, as well as the glycosylation. So this particular protein has two N-glycosylation sites with a very interesting kind of glycosylation. This was new to us before working on this. In Pichia, many proteins are produced with high mannose glycans, which also feature these phosphodiester's, so phosphorylation inside of the glycan. So in order to understand at the intact protein level what we have, we used a combination of peptide mapping, where we can see individually the N-glyco profile at each site. Then we use deglycosylated intact mass to study the truncation, which is happening. And then we, of course, look at the complete proteoform profile in the untreated intact mass. So for the mapping, we use a variety of softwares, but in this case, we found that Proteome Discover and 2.5 with the Bionic node allowed us to perform the identification of these phosphoglycan peptides. And of course, we use the Menora node for label-free quan in order to put this together. We curate the data manually. That is, we combine all the charge states present for a specific peptide sequence and then calculate a relative abundance for each of these forms. Something like this, where we have a differing degrees of hexNAC, and well, it's the same core really, but differing degrees of mannose here, hex, and differing degrees of phosphorylation. And each of these sites have their own unique distribution of glycoforms. When we move on to the truncation analysis, of course, using our full scan PTCR MS technique, we're producing these beautiful, I think, intact mass spectra of the truncated species. And it's complex, right? There's actually quite a bit going on here. We were able to identify 9 or 10 of these, the more species, but not all of the abundant species by accurate mass within a window of 50 ppm. It would be, and they these data largely match what's happening at the raw data level. So that's great. We believe in the deconvolution, but the identification of some of these species that we can't find would be made better. It would be a vast improvement over just using MS1 or intact mass to identify these species if we could use top-down to improve this. This would also help save time and give us better confidence. So we spent some time trying to generate top-down data from these charge-reduced intact mass species, which we do these experiments using high pressure from 8 to 20 millitorr. When we perform a more standard MS1 analysis using low pressure, of course, in this case, it's 0.2 millitorr. This is the spectrum that we get. We like the charge-reduced spectrum because this helps with deconvolution and confidence at that intact level. However, what we found is top-down really likes the low pressure. And so that's where this has been essentially done for many years now, and is now being developed further using PTCR at the MS-MS level. A lot of this work has come out of Luca Fornelli's lab, and they've done a great job with that. So basically, at this point, we still think top-down is best done at low pressure. So we did some experiments at low pressure, and we're using our detailed mode in a 20-minute method. And so we have about 2 to 3 minutes of analysis time here. And this is, I would not consider this an optimized experiment, but you can see how many scans we're getting, even with, we're doing 15K, but 10 microscans at 15K. And we're also doing 120K CID MS-MS and 10 microscans there as well. And so this can take quite a bit of time. We were initially hoping to generate very high quality data, and we were interested less in the number of proteoforms. And in this case, we're happy with where we're at in development. Seeing is that we can perform this in an unguided manner. This is just DDA, and we've produced in essentially our first try doing this confident data for 4 of the species that were identified by the intact mass results, but they're now automatically identified using MS-MS here. So this is really promising. We like this. And there's a lot of room left for optimization. Okay, and then in the final example here, we have the untreated mass, where this is a single charge state now of the untreated 25 kilodalton glycosylated, phosphoglycosylated species. And when we deconvolve that, I can show you that the deconvolved profile largely resembles what we see in this single charge state in the raw data. So that's good. That gives us some confidence that the deconvolution has been done correctly. And the red dots here, aside from the ones in the baseline, the red dots all correspond to the single most abundant truncation isoform, where we found a little over 20 different glycoforms for this. So it's a complex species, and all of these unlabeled species are the other truncation isoforms, each with their own glycosylation. All right, so then in the very last part of this, we recently demoed the BIOS software, which allows one to do the reconstruction of the data that is acquired at the peptide mapping level, and this can be superimposed on the deconvolution results. And so in this case, we used simply the top 3 now, truncation isoforms, And each of these are reconstructed using the combined glycoform profiles. So the potential combination of every glycan here and every glycan here with the top 3 truncation isoforms. And by first glance, I think that this is a pretty nice fit. One of the things that we're not able to do in this, and so if there's somebody who's savvy with BIOS who can tell me how to do this, that'd be great. But we haven't figured out how to adjust the relative abundance of these different truncation isoforms, which certainly has an impact on the way the reconstruction looks. So here's just a zoom in. There are some unaccounted for species here, but of course, we only went to the top 3 of the truncation isoforms. And so doing that in a more complete manner in the future is also interesting to us. And I'm going to leave you with that. I really appreciate the questions today, or the attention today, and in your questions in advance. I thank my fantastic colleagues, Jennifer, Lee, Trevor, and of course, Dr. Russell, and our collaborators at Thermo. Particularly Roman was very helpful in helping us get to this point, and our other folks, collaborators at UTMB. So with that, I'll take any questions you might have.
Great, thank you, Aaron. That's fantastic stuff. Very nice to see such a rigorous approach to method development. It's you know, the intact mass is definitely something a lot of people can benefit from. You know, as we've seen often in the past that, you know, people worry about top down, but sometimes getting the top is even the challenge, never mind the down part. So this is great. I just had a quick question. I mean, I know you've developed this around the Orbitrap platform, but do you see this as potentially being applied to other platforms as well, or parts of this approach? Could it be extended to another type of platform, do you think?
Absolutely. Yeah. Well, so we saw the data from the Matthew Bush lab, and actually in that case they call this Captor. It's similar technology, PTCR, and Captor are, I mean, more or less the same type of approach. We're using an anion to reduce the charge state of intact proteins. Did that answer your question? I think the thing that makes it very usable, maybe to add on to that in the Orbitrap, is the ability with the ion trap to do MS to the N, right? And this can be applied now to any level of tandem mass spec that we would want to do. And then I think, you know, the data I showed hopefully generates some questions and some understanding also about how the different pressure regimes and the way that you kind of have to commit initially to that experiment and can't be digitally changed in the moment. So there are some limitations and advantages.
Great, thanks. So if other folks have questions, you can raise your hand or to speak live or unmute yourself, or you can just drop it in the chat. But we do have a couple in the chat. There's a question from Mike Neerman. He says, does lowering the charge state of denatured protein reduce the CCS?
Yes, we think it does. Now, our data did not show that specifically, but yeah, I'll go back to that example from the Bush lab. I really like it a lot. And in this case, yeah, so the CCS, which is shown as omega here, this shows us very specifically, and actually there's data in the Sanders paper from the Broadband lab that says virtually the same thing, that native and kind of lower charge states that you get from native MS exactly correlate with this low collision cross section area. And then as you kind of extrapolate further through the denatured and then the denatured reduced supercharged BSA, those high charge states correlate with a very large CCS.
Yeah, my question was, could you take the reduced material and then move it back down in collisional cross section just by reducing the charge, or do you need the disulfide bonds to keep that intact structure?
Yeah, so that's what we think that we're doing. We don't have ion mobility built into this system, but we do think that in this case, we have, I'll go back to the big example here. In this case, we do think that what we're doing by reducing the charge state allows us to get basically better data. And this changes the transient decay. So I think that's the source of the improved signal. And that's also the case for this protein as well, in a much more exaggerated fashion. You know, we're well over the 100+ charge state regime in this large floppy intact protein. And then by reducing this in charge, I think we're doing a lot more than just changing the charge state. I think we're simply collapsing the CCS as well, though we don't have the analytical equipment to show that directly.
Okay, thank you. Interesting.
Here's another question from Julian Whiteledge. Says, very nice. Do you ever use high resolution for intact mass measurements?
Yes, we do sometimes. This is kind of helpful when, very helpful when we have, I guess, you have this intersection of micro heterogeneity and macro heterogeneity. We've run some histone samples here, really just as tests. That's not like a thread of research that we do, but I mean, as an example, I think a lot of folks are familiar with histones, and you know, it's almost trivial to get isotopic resolution on a histone in conventional mass spec, you know, with a scan range of 2000. But when you have several histones eluting at the same time, and it can get very messy very quickly. And so there's an example where we see the charge reduction could be very useful for producing much cleaner intact mass spectra there. I think with histones, there's a desire to do top down. And so that's where we get into this problem. I'm going to go very quickly through this, that we find very interesting. This kind of, I guess it's a hard break between what we're doing to produce very high quality intact mass data, and then the way that top down is really has to be performed inside of the Orbitrap. And so we have to, you know, take a step back and then do those experiments, say for the histone experiment.
Great, thank you, Aaron. Are there any additional questions or things people want to comment? Questions for Aaron?
I've got a quick one. On the last slide that you were showing on the initiation factor 2, could you go back to that one?
Yep. Yeah, let's get here. There we go.
That, right there. All right. I think that's sweet, the way that your mass accuracy gets so much better when you do the charge reduction, and it really moved the large peak over. The peak just to the right of it didn't move at all. Do you believe that that is a modification on that protein, or is that something totally different?
That's a great question. Actually, we looked at this a little bit, and the mass accuracy actually does shift quite a bit.
Oh, it does.
I'm not showing that here. It's roughly, I mean, hundreds of ppm, roughly the same as this. But we see it twice, and we see it with greater fidelity in the bottom result. As a matter of fact, if you zoom in, this little nugget here right next to it also shows up with kind of greater confidence, I would say, in the deconvolution results. So it does. Everything does improve. We actually have no idea what this PTM is here, though. That's the catch. We didn't make it that far.
Okay, I've just, you know, straining looking at the tick mark, but beautiful stuff. Nice.
Thank you, Mark.
Anyone else have a question or comment or a good joke? If not, then I'd like to thank Aaron for this wonderful tour of this PTCR method. You know, it's a really nice, rigorous study here, and it looks like it's going to be a really useful tool for a lot of folks going forward. Oh, wait, I see a couple hands. Chris Brown has a question.
Oh, I was just saying thanks.
Oh, okay.
Sorry.
All right. Yeah, just some claps.
Yeah.
All right. So again, thanks again, Aaron.
you,
We
Chris.
appreciate it. And just so folks know, June 23rd, we'll have our next one. Will be information coming out on that shortly next week or so. And that's it for today. Hopefully we'll see some of you in Minneapolis in a couple of weeks. And thanks again, Aaron. And I wish you all a nice day. Thank you. Bye-bye.
Thank you.